G'day everyone, hello and welcome to another live stream of It's All Happening at UTAS. I'm Jamie Chapman, this is Dr. James Crane.
Hi everyone.
And today we're going to be looking at respiratory physiology predominantly. But if you have any other questions related to the respiratory system, we're here to help. Dr. Crane will be taking most of this session, but I'll be watching chat, and please remember, if you're here, come say g'day. We're happy to help answer questions that you may have. Otherwise, we'll just rabbit on for a while about the respiratory system. So I'm going to move off to the side. I'm going to put you in the very capable hands of Dr. Crane, and yeah, please feel free to come along and ask questions, and we'll try to help you out. Okay.
Excellent. Thank you, Jamie. So I wasn't here last week, but Jamie tells me that there was a question looking or related to the difference between obstructive conditions and restrictive conditions, right? As it refers to lung function and how the lung function is actually going to be affected in those different conditions. And I think someone brought up the idea that COPD, so chronic obstructive pulmonary disorder, or diseases, seem to have elements or features of both of those. And that's sort of true, but it's sort of also not true. And if we, and so the COPD conditions are definitely referred to as obstructive disorders. So what I thought we would do is, first of all, we'll have a quick look at this diagram here. So this is just off Wikipedia. And it gives you a pretty clear idea of what we're talking about when we're talking about these different cycles that go on through here. And I have my little pointer, so that's working out really well. Okay, so if we're just sitting at rest, breathing in and out, all right, we have this nice little variation in the volume of our lungs, right, the amount of air inside of our lungs. And so we can see that what's happening is that as we breathe in, it goes up to about 37 mils per kilogram, and then it comes back down, and then comes back down. So that's just a nice, easy breathing rhythm that would be happening at rest. And so that's what we call our tidal volume, right? So this is just our nice sort of tidal volume that's going on here. But we all know that we have the ability to breathe, to have larger changes happening inside of our lung volume. And we can do that by simply doing things as simple as taking in a big deep breath. So, taking in as big a deep breath as possible, obviously we're going to exceed this inspiration that occurs as part of the tidal volume here. And we're going to go up to what becomes our maximum lung volume. And so the difference between the tidal volume and what our maximum lung volume is what we call our inspiratory reserve volume, right? And it sort of is what it says it is. It's the reserve volume, the extra volume that we can take and make use of on when we're inspiring, when we're breathing in, above and beyond what we would normally be using just at rest when we're sitting there, calmly relaxing. Okay, so if that's our inspiratory reserve volume, then when we breathe out with force, so we breathe in, and we breathe out completely, we end up with this, what we call our expiratory reserve volume. And so that extra amount of air that you're able to breathe out above and beyond what you would normally be breathing out in your tidal volume is what we call our expiratory reserve volume. Now, something that is on this diagram that you may not necessarily be aware of, and it's certainly not something that you think of normally, right? We're quite used to taking in a deep breath, and we're quite used to taking out and breathing out. And you're used to thinking that we have, we've breathed out and we've gotten rid of all of the air of our lungs. All of the air in our lungs was breathed out forcefully. But it's not actually true, right? What we have is that there's a little bit of air that is always stuck inside the lung tissue, right? And that's what we call our residual volume. And I was just talking to Jamie about this earlier, right? And so what this relates to is that once we get into the bronchioles, and I know Jamie talked to you about this a little bit last week, once we get into the bronchioles, we start getting into bronchioles that have a diameter of about 1 millimeter or less, and they lack the cartilaginous rings, or the segments of cartilaginous rings, to help maintain their diameter and to keep them open. And so what happens is, if you think about what's happening during a breathing cycle, we breathe in, diaphragm contracts and it moves down, chest cavity, especially on a big inspiratory reserve volume breath, or the chest cavity is going to increase, and so we decrease the pressure in the chest cavity, and that's going to be causing the air to flow in through the trachea and into the lung tissue and into the lungs, right? When we breathe out, the opposite happens. Diaphragm comes back up into its resting position, and the intercostal muscles and the chest wall muscles contract and the abdominal muscles contract, and we see a massive increase or an increase in the thoracic cavity pressure. And that increase in pressure then causes the pressure inside the lungs to exceed the atmospheric pressure, and so the air flows out. But what that increased thoracic pressure also does is it exerts pressure on those very narrow bronchioles. And so at a certain point, so when it first starts, there's enough air leaving those bronchioles and moving through those bronchioles to resist the pressure that's being exerted on them. But as the amount of airflow decreases, eventually you get to a point where the thoracic pressure causes those bronchioles to close over. And they close over before you've been able to get all of the air out of the lung tissue. And so this causes this part of this volume to be trapped inside the lungs, and this becomes our residual volume here. Okay. So let's have a look at some of these other things here, and I really want to particularly focus on this vital capacity, right? So this forced vital capacity is normally what you would see if someone's actually sort of a big deep breath in and a big deep breath out, right? A big exhalation. So this vital capacity, if you look at how this stretches, it stretches from the entire maximum volume that you can obtain at the peak of your inspiratory reserve volume, and it extends all the way down to the bottom, or the maximum amount of the expiratory reserve volume, so the maximum amount that you can exhale. And so these values here, especially the forced vital capacity and the expiratory reserve volume, are going to become really important as we start talking about, and also I should say the inspiratory reserve volume, as we start talking about obstructive and restrictive airway conditions. So let's first of all talk about those obstructive airway conditions. So obstructive airway conditions are things that are going to narrow the air passageways, right? So they're going to narrow the bronchi and have potentially effects even at the level of the bronchioles, depending on how severe the condition is, right? So things like asthma, even cystic fibrosis is considered an obstructive airway condition as well, because of the mucus that builds up inside the bronchioles. And so what's going to happen is that the amount of, so you can breathe in. Breathing in is maybe a little bit more difficult, but mostly you can actually get the air to come in. That's not as big of a problem. It's actually exhaling that becomes the problem here. And the problem is that because the passageways are narrower, it limits the amount of air that can actually pass out of those passageways. And so therefore, because it's limiting the amount of air that can pass out of the lungs, the volume of air traveling through those very, very small and unsupported bronchioles is reduced more quickly. And as a result, we see them collapsing under the force of the thoracic pressure, the increased thoracic pressure that you see with exhalation, right? And so what you'll see is that you'll actually see that as someone breathes out with this, so they have this thing called FEV1, which is forced expiratory volume within 1 second, within the first second of first breathing out, right? And so someone who has a chronic obstructive pulmonary disorder or one of these obstructive conditions will see a decrease in the volume of air that they're able to breathe out in that first second, right? And that's largely because the residual volume actually will increase a little bit because those small bronchioles are collapsing more quickly and more readily, and it's limiting the amount of air that we're actually able to get out of the lung tissue. So when you're diagnosing someone with an obstructive condition, they actually use this thing which is a ratio between the FEV1, which is sort of a measure of this expiratory reserve volume, so the forced expiratory volume within 1 second, and they put that over the entire vital capacity, right? And so the anything of a ratio of about 0.75 or lower is considered to be indicative of an obstructive airway. So that's basically saying that the FEV1 has decreased, but our forced vital capacity has not decreased as much as the FEV1, right? So what happens is that initial ability to get rid of that air in that first second decreases. But you may still have this slow leakage of some of the volume out of the lung tissue over a longer period of time. And so the vital capacity doesn't change quite as much as the FEV1 does, the forced expiratory volume in 1 second. So how does that then differ to a resistance problem, right? So was that restrictive? I knew that I'd missed that word, right? So how does that differ from a restrictive problem? So a restrictive problem, when we're thinking about restriction, we're not really talking about the restriction of airflow so much. We're talking about the restriction of lung expansion, right? And so the actual compliance, if you will, of the lung tissue. And so conditions where you have scarring and the buildup of scar tissue and connective tissue inside the lung tissue itself, not necessarily inside the tubes, inside the bronchioles and the bronchi, but actually inside the tissue itself, can limit the ability of the lung tissue to expand. All right, and so therefore I take in a deep breath, or I try to take in a deep breath, diaphragm drops, chest cavity increases, the pressure decreases, the air tries to flow in, but the lung tissue itself has a limit to how much it's able to expand, right? And so often that's related to infections, other things that have caused some sort of scar tissue that are occurring. But, I think Jamie actually possibly mentioned, early on, one of the things that you need to do is produce surfactant because there is a considerable amount of surface tension. So one of the advantages that we have by having this sort of massive alveolar bed inside the lung tissue is this massive increase in surface area that allows us to exchange gases really, really readily, carbon dioxide out and oxygen in. But all of that large surface area, right, is coated in water, right? It's coated in fluid. And because of that large surface area, we actually end up with a fairly large amount of surface tension. So surfactant is your detergent type substance that is going to break down or at least weaken that surface tension and allow the lung tissue to expand. But without that surfactant, the lung tissue isn't going to be able to expand as much. And so that becomes, again, one of these sort of restrictive conditions. So what's this going to do for our diagram here? Well, what's going to happen is that we'll actually see a decrease in the vital capacity, and we'll see that decrease predominantly because our inspiratory reserve volume is going to be decreased, right? There's actually going to be a limit to how far our lung tissue can expand and how much air we can actually bring in to our lungs because it's not able to expand quite as much, right? So that inspiratory reserve volume will decrease, and you actually probably, because of the reduced sort of spring, I guess, and elasticity of the lung tissue, you probably also see a fairly equivalent or not too dissimilar decrease in the forced expiratory volume in 1 second. And I guess that's also related to the fact that if the inspiratory volume is going to be less, then the amount of volume that you can expel within 1 second is going to be less. And so if you look at the ratio between the forced vital capacity, so if you put FEV1, the forced expiratory volume in 1 second, over the forced vital capacity, in a restrictive condition, you actually end up with not much of a change from a normal situation. So they actually end up being fairly similar or fairly close to 1 because they actually because they are both sort of shifting to the same degree. Whereas in an obstructive condition, you're seeing a much greater change in the FEV1 than you are in the forced vital capacity. And so if we're thinking about things like emphysema then, so what's happening in emphysema? So emphysema, we're seeing a breaking down of the elasticity and the elastic fibers inside the lung tissue, where we're limiting or reducing the amount of elastic recoil that we have in that lung tissue. So I can take in a nice, big, deep breath. There's very little resistance to the flow of air into the lung tissue. But because I don't have that elastic recoil, the amount of air that I'm able to, or the force with which I'm able to get the air out, and the volume of air I'm able to shift within 1 second is going to be massively reduced. And so we'll end up with a reduction in our FEV1, which means that we'll see a reduction in our expiratory reserve volume. And so this also then becomes one of those chronic obstructive pulmonary diseases or disorders, because of the fact that you're effectively getting a reduction in FEV1 and a collapse of those small terminal bronchioles, so therefore sort of an increase in that residual volume. So how's that gone, Jamie? Has that answered things? Hopefully that's answered the questions, and we've had some feedback on the chat. No feedback on the chat? Is anyone out there? Quiet. Is anyone out there?
We currently have 12 people watching, but they're quietly watching.
Oh, that's good. Okay. So what else can we talk about?
So I know that there were a few questions not asked of us directly, but certainly on the chat, and I know I'm asking this without notice. So if you're not able to answer, I completely understand. But people were having a few questions regarding the hemoglobin.
Oxygen dissociation curve.
Hemoglobin affinity.
Yep.
And the shifting that takes place to the curves and things.
Sure. Okay, so we might need to grab I wonder if we can find an oxygen. Let me just see. Oxygen dissociation curve. Look at that. Google providing all of the best things for us. Let's have a look at that. Well, that's worked out really, really well. Okay, so this is actually one of the things I think that got me into really wanting to pursue physiology. I remember getting these lectures when I was in undergrad and just thinking that it was so beautifully set up, that it was such an incredibly elegant system to maximize the delivery of oxygen to the tissues at the point and at the places where it really wants to see that oxygen, right, where it really needs it. Okay, so if we think about hemoglobin, right? So hemoglobin is a complex molecule. It has 4 units to it, and each of those units has a ferric compound, an iron compound, and as a result, it's able to bind 4 oxygen molecules, right? So each hemoglobin molecule can bind 4 oxygen molecules. What's intriguing about this, and what's important and interesting about this, is that the affinity that the hemoglobin molecule has to those 4 oxygen molecules changes as those oxygen molecules get added to it. So if you consider that, when we have just one oxygen molecule, it binds to that with a really, really high affinity, which affinity means that it's going to really strongly attach to that oxygen molecule. And as we go from 2, the second one is bound with a high affinity as well. And then the third one, it's a little bit less strongly bound. And then the fourth one is even less strongly bound again. And so they're not all attached to that hemoglobin with the same strength. And so what this means is that we can lose different components or different numbers of those oxygen molecules from that each hemoglobin molecule at different times, or related to different situations. So the fourth one will come off first, then the third one will come off, then you might get the second one to come off, and it's actually quite rare for that first oxygen molecule to drop off completely. it needs to have very, very specific conditions for it. So if we look at this oxygen dissociation curve here, if we're looking at the blue one here, this dark blue one, I'm just trying to understand this curve just to make sure I've got it right for us. Okay, so if we assume that the blue one is our normal situation, right? So this is our normal oxygen dissociation curve, and we haven't really done anything to change that. So these sorts of things here, we're seeing changes in pH, DPG, you don't quite know what that is, temperature, and we've got changes in pH and temperature here as well, right? And so we're going to sort of look at those two situations. But if we're looking at just a normal situation where none of those things have really changed very much, what's this curve saying? Well, it's saying that at a partial pressure of oxygen of 100 millimeters of mercury, so where we have a really high saturated oxygen concentration in the bloodstream, then we have basically as close as we can get, right, to almost 100% of our hemoglobin molecules bound onto oxygen, right? So all 4 of those binding sites are completely bound onto oxygen. Now, as the oxygen concentration decreases, you see that we start to see the binding of oxygen, or the amount of oxygen bound to hemoglobin, decreasing, right? And so it starts off slowly, so we might see maybe just a few of those hemoglobin molecules losing some oxygen. But once we get to around, say, 50 millimeters of mercury, so about halfway, we see a pretty rapid decrease in the oxygen dissociation, right? So the ability of oxygen to disappear and come off that hemoglobin molecule. And this is what really made me very interested in this when I was in undergrad, right? So if you think about this, where am I going to see really, really high oxygen concentrations? Well, I'm going to see high oxygen concentrations in the lungs. Breathing in, bringing in oxygen. So as the blood, the oxygen, the hemoglobin moves through that tissue, it's going to encounter a high, very high partial pressure of oxygen. We're going to see almost a complete binding of the oxygen to the hemoglobin. So all of the hemoglobin molecules that are leaving the lungs are going to be fully bound to oxygen. Now what's going to happen? It's going to go into the left atria, into the left ventricle, and it's going to get pumped all the way through the body. And so as it's being pumped through the body and as it's moving through the body, it's increasingly encountering tissues that have lower and lower levels of oxygen. And that makes sense because the tissues are out there using oxygen, right? The oxygen is diffusing into the cells, it's being used by the cells in metabolism, and carbon dioxide is coming out as a waste product. So as we move through the body, the concentration, or as you get further and further towards the tissues, the concentration of oxygen inside the bloodstream is going to be decreasing. And so as we move to these concentrations of oxygen where the oxygen concentration in the tissues is decreasing, so around 50% of what it would be in the lungs, or down around 40% of what it would be in the lungs, then you start to see the hemoglobin molecules really starting to release their oxygen, which is exactly what you want them to do, right? You don't want them releasing their oxygen straight after coming out of the lungs. There's plenty of oxygen in the bloodstream already. There's plenty of oxygen floating around. So what do we actually want? We want the oxygen to be released from the hemoglobin as it gets to tissues that have lower and lower levels of oxygen already, right, and around them. And so we can actually see that what's happening is that we have this drift, or this rapid sort of reduction in the binding of oxygen to hemoglobin as we get into an area where the tissues need that oxygen, right? So this encourages the oxygen to come off of the hemoglobin, enter into the cells to be used for metabolism. Obviously then this deoxygenated hemoglobin makes its way back to the lungs, becomes reoxygenated again, and the whole cycle continues. Okay, so how does this then relate to these other curves? So we've got this green curve here. Well, let's focus on this red curve here. Okay, so if we're looking at this red curve, So if we're saying that at about this point here, hopefully you can see this on my diagram, right? So this is our oxygen hemoglobin saturation. So we're sort of at about 82% of the oxygen hemoglobin saturation. So on the blue line, which was our normal situation, in order to get to that point, right, of oxygen saturation of around 80%, we had to have a partial pressure of oxygen of about 50 millimeters of mercury. If we look at the red line, what we've done is we've shifted that whole curve to the right. And so now, if we're looking at it, we can get to this same level of oxygen dissociation. So about a saturation of about 82%. So we've taken about, 18, or, it's not quite at 100, but somewhere around there, of the saturation has decreased. We can get to that point, but now we don't need to be at 50. We can actually get to that point at 60%. So we can release the same amount of oxygen, but at a higher partial pressures of oxygen in the tissues. So what is it, the things that are causing this? Well, we're seeing an increase in temperature causes this shift to the right, and a decrease in pH. And I don't know, have you been able to work out what DPG is? Someone may know on the chat. I just didn't look at the things. I don't know what the acronym stands for. But if we just focus on temperature and pH, what's going to happen when you're exercising? What's going to happen in the muscles? What's going to happen in the tissues when they're working and they're using more oxygen, right? So let's just focus about exercise. We're exercising. We know that we're going to be using more oxygen. There's going to be oxygen coming in to produce the ATP that we need to generate the contraction of the muscle fibers. So what's going to be the outcome of that? Well, the outcome of that increased metabolism and the increased movement is going to be an increase in temperature in the tissues. But we're also going to be producing lactic acid as a byproduct. We're going to be seeing and also carbon dioxide, right? And so through the carbonic anhydrous cycle, we'll also see an increase in carbon dioxide inside the tissue, which will shift that equilibrium potential or equilibrium of that reaction, so that we'll end up with more hydrogen ions inside the tissue and inside the bloodstream. So when we're exercising, we would expect to see an increase in temperature locally and a decrease in pH locally, so that it becomes a little bit more acidic. And these things are going to shift that curve to the right. And so as a result, we'll be able to release more oxygen for a given, or we can release that 80, we can get to about 82% of saturation at 60 millimeters of mercury as opposed to 50. And so that would make perfect sense. We are exercising. The muscles need more oxygen. We don't want to wait until the partial pressure gets down to 50 or 40 before we release that oxygen and provide that oxygen for the muscle tissue. So this is a mechanism for the hemoglobin to sort of realize, and obviously it's not conscious, but there's this sort of realize, or this response to these changes in pH and the response in the changes in temperature, that is an indication that the muscles and the tissue is doing more work and could do with some more oxygen. And so it will release that oxygen more readily. And then the converse is true. We have a decrease in temperature, we have an increase in pH, right? And so what we'll see, let's just focus on the decrease in temperature. So just a simple decrease in temperature, we're going to see a slowing down of enzymatic reactions. We're going to see a slowing down of the need for oxygen and the energy that's going to be produced by respiratory, cellular respiration, right? The TCA cycle.
So Ben has asked, and I think you've probably already answered it through this description, is when we exercise, hemoglobin releases more oxygen. So
yeah, exactly right. So when you're exercising, the reason it releases more oxygen is because part of the byproduct of that extra work is an increase in local temperature, decrease in local pH. There's also changes in carbon dioxide. So carbon dioxide can affect that. Obviously, the tissues themselves are possibly using more oxygen, so they might be more likely to get to areas of tissue which you actually have lower partial pressures of oxygen, and so that's going to cause the oxygen to form off the hemoglobin more readily. Right, and so I think the most important one really is this red curve, which is saying that, look, we can shift this to the right and we can increase the release of oxygen or the rate at which oxygen dissociates when we need it to when we need more oxygen in the tissues more readily. Obviously, if we decrease our temperature, we can go the other way. We shift it to the left, and now we need to get down to about 40 millimeters of mercury of partial pressure of oxygen to get to that same oxyhemoglobin saturation point. So we need to actually have less oxygen inside the tissues before we'll actually release the oxygen off the hemoglobin as much. And this sort of plays a role in the effect of cold to delay damage associated with respiratory issues and a whole range of different things, right? Is that the cold is going to slow down enzymatic reactions, and it puts as a result, there's far less demand for energy and far less demand for oxygen. So hopefully that's answered that question.
I believe so. So just so you know, DPG stands for 2,3-diphosphoglyceric acid.
Oh, okay. I'm not familiar with what that is, so hopefully maybe someone else knows.
It's on red cells, apparently.
Right, okay. It might be an internal regulator of red blood cell function. Yeah.
Yeah, so it helps to control the ease at which oxygen is released from the hemoglobin.
Oh, okay. So same as the temperature and okay. Lovely. All right.
Ben says yes, thank you very much. It does answer his question.
Nice. What else can we talk about? Respiratory function.
So DPG decreases oxygen affinity.
Right. Yeah.
No, that was a question.
Oh, right. So, well, it sounded like what Jamie said. That it increasing levels of DPG made it easier for oxygen to come off the hemoglobin. Yeah. What else do we like to talk about? I don't know. Any other questions out there, anyone? Lung respiratory physiology it seems more complicated than it is, I think, sometimes. Effectively we're just dealing with gases moving in and out, following Boyle's law, following this, that gases will move from an area of high pressure to low pressure. And when we're breathing in, all we're effectively doing is just changing the pressure in the thoracic cavity, and therefore, adjusting the pressure inside the alveolar to being, and the lung tissue to being below atmospheric pressure or above atmospheric pressure. All right. So we could.
What about control of breathing?
Oh, okay. In terms of brainstem related function.
Yeah. So the homeostatic mechanisms with control of breathing, receptors, control center effectors.
Okay. It's not something that I've got on the top of my head. Okay, right. So what I can tell you about that, right, is that so it's actually all to do with acid. It's actually all to do with pH. So and this sort of relates to that. Hopefully we've done this a fair bit. Let's just get my little pen tablet going here. Gonna need that. All right. Excuse me for a sec. Okay. So it actually relates very much to this carbonic anhydrous cycle. Carbonic acid cycle, right? So this is interesting. So this is basically what's happening. We have if we have H2O, and then we have CO2, carbon dioxide, and we have this bidirectional arrow here. We first produce this compound called carbonic acid, H2CO3, right? And if we look at this, we've got H2, got an oxygen here, we've got 2 oxygens here, so we're going to have 3 oxygens and a carbon. That's a pretty unstable molecule. And so the first thing that's going to happen, this actually really wants to dissociate either back into water and carbon dioxide or into bicarbonate, HCO 3 minus, right? And hydrogen ions, right? And so hopefully you'll remember from just simple kind of chemistry that whenever you're seeing an increase in free hydrogen ions, and okay, in water that might actually be these H30 plus molecules, but I usually like to just sort of simplify things and talk about free hydrogen ions. So if we're talking about free hydrogen ions, whenever we see an increase in free hydrogen ions, then we're seeing a decrease in pH, right? So pH is decreasing as hydrogen ions are increasing, right? And so as a result, this is, what we say it's becoming more acidic. So if we think about what's happening here, when the cells are working, when they're burning oxygen and using that oxygen to produce ATP, then the byproduct is carbon dioxide. So we're going to see a buildup of carbon dioxide inside the cells, right? CO2 is going to be inside the cells. That's going to create a diffusion gradient out of the cells into the tissues and then into the bloodstream. So we're going to see an increase in carbon dioxide inside the blood vessels. That increase in carbon dioxide is going to shift this reaction in this direction, right? So the reaction is going to want to move in this direction. So that's going to create the production of this carbonic acid, and that will rapidly break down into bicarbonate and hydrogen ions. And so effectively, whenever you're respiring and whenever we're producing carbon dioxide, we're making the blood, the local concentration of the hydrogen ions in the blood a little bit higher, and the pH is decreasing, and so everything's becoming a little bit more acidic. So in situations where you are not breathing as much, what's actually happening is that you are not breathing as much, so therefore we're not releasing that carbon dioxide into the atmosphere. So the carbon dioxide inside the bloodstream builds up. We see an increase in hydrogen ions. We see which results in a decrease in pH. And so that's what we call acidosis. And this can become a really big issue, right? So this is a clinically very, very important thing. All right. So if you think about it, if you've got a patient that's not breathing, or if you've got someone who hasn't been breathing, they're not blowing off carbon dioxide, but their cells are still using oxygen, right? They're still alive. They're still using oxygen and producing carbon dioxide. So we're seeing, with someone not breathing, we see an increase in carbon dioxide. We see a shift in this reaction to the right, and as a result, we get an increased acidification of the fluid inside the blood vessels. The blood, right? I don't know why I called it the fluid inside the blood vessels. Acidification of the blood, which is what acidosis means. This can become a problem because a lot of enzymes actually require very, very specific pH ranges. And if we get outside of those ranges, if we become a bit more acidic, they can become denatured and they can cease to function properly. So you can have really long-term effects of this acidosis. So one of the things that, is really important is to get people to breathe again. And not just to bring in the oxygen that the cells need, but to also begin to blow off this carbon dioxide so that we can reduce the carbon dioxide levels and hopefully bring this acidosis back down and get back to our sort of neutral pH of 7.35 to 7.45. So just before I move on to talking about the respiratory control centers, this becomes really interesting for things like COPD, like chronic obstructive pulmonary disorder. So we just talked about these disorders. So these are disorders where there is difficulty in breathing out, right? They have difficulty in expelling air. And if you think about the air that we're trying to expel, that's going to have a high concentration of carbon dioxide. So in a patient that has difficulty breathing out, they are at a higher risk, obviously, of developing acidosis because they're not able to clear carbon dioxide as much. So this becomes an inherent problem and something that needs to be dealt with, but it becomes a really significant issue if they ever need to be treated for hypoxia. All right, so if they have a situation where they're becoming hypoxic, it becomes really, really difficult because you have to be very, very careful giving them oxygen, right? So you can't give them a very, very high concentration of oxygen because they'll breathe in that oxygen. The cells will go, yay, fantastic. They'll start using that oxygen like gangbusters, producing a lot of carbon dioxide. But this COPD patient has trouble expelling that carbon dioxide. So you can really rapidly send them into an acidotic state and cause a lot of problems. So, a lot of times people with COPD will wear a bracelet to let people know and let paramedics and emergency staff know that they do have COPD, so that they know that they need to be very, very aware of the oxygen that they're administering to this patient. Okay, so how does this relate to the neural control or the respiratory control centers? Now, the one thing I can't tell you off the top of my head, and I feel bad because I'm a neuroscientist, is exactly where those control centers are. I know they're definitely in the medullary region, but I can't remember off the top of my head exactly what they're called. But if we're looking at this, what it is, so people sort of think, oh, well, I breathe when I don't have enough oxygen, or I'm not, I need more oxygen, so therefore I'm breathing more. It's actually not the oxygen that's driving increased respiratory rate. It's the increase in carbon dioxide inside the bloodstream, which is causing an increase in hydrogen ion because of that carbonic cycle. And so it's actually the decrease in pH that is causing your increased respiratory rate, because we have acid sensing neurons, right? So these effectively detect and have receptors that detect free hydrogen ions. They detect a change in pH in the bloodstream, and through a process, a neural process, they send signals out to the diaphragm that increases the contraction of the diaphragm and increases the respiration rate. The idea being that by increasing the respiration rate, I can blow off carbon dioxide, reduce the carbon dioxide levels, get my hydrogen ion levels down to normal, bring my pH back up to 7.35 or so, to the normal range. And now, because that pH is fine, the acid sensing neurons don't become activated, and we can go back to our, we can drop our respiration rate when everything returns back to normal. So it's a key element of respiratory function that it's not actually oxygen levels that are driving the changes in respiration rate. It's actually carbon dioxide levels through the production of hydrogen ions and the production of acid sensing neuron and the sensing of that change in pH by acid sensing neurons. That carbonic anhydrous cycle is particularly interesting. So Jamie, feel free to jump in if anyone wants to change anything, but I'm just going to keep banging on about this for a little bit. So I really like this. It's a really simple equation, right? But it explains a lot of different things. So what are some interesting things that this explains? Well, one of the interesting things that it explains and probably conveys to you is that I think something in the order of 70%, at least 70%, if not higher, of carbon dioxide that's carried in the bloodstream is actually carried in the form of bicarbonate ions. So there is some carbon dioxide bound onto the hemoglobin as well, but the vast majority of carbon dioxide travels through the bloodstream as part of this bicarbonate molecule, right? So the carbon dioxide joins onto the water, produces this bicarbonate molecule. This bicarbonate molecule is then taken to the lungs. And so what's going to happen when we get to the lungs? Well, we're going to see an area, if we're breathing properly, with reduced carbon dioxide, because we've been blowing the carbon dioxide out. So that area is going to have reduced carbon dioxide. That's going to shift the equation back to the left. And then that will produce carbon dioxide. So the bicarbonate will get turned back into carbon dioxide and water, and will blow off the carbon dioxide, right? So that will then be breathed out. The carbon dioxide levels will decrease, and then the hemoglobin will bind onto the oxygen. So this whole pathway, this carbonic acid cycle, is the principal way by which we carry carbon dioxide around the bloodstream. And it makes a lot of sense, right? It actually works really, really well, and it doesn't take too much to actually see how moving from one side to the other can have a pretty significant effect on things. The last thing I want to sort of point out is, you've probably all heard that the oceans are becoming more acidic, that increased carbon dioxide in the atmosphere is causing the oceans to become more acidic, and gradually we're changing coral, and all sorts of marine environments are actually being affected. And so, you may have asked, or you may have been asked at barbecues, or heard people debating this thing, talking about what's going on, right? And how could that possibly be? And it all comes down to this equation, right? As we have seen an increase in carbon dioxide in the atmosphere, carbon dioxide is going into solution into the oceans, it's binding onto the water, producing this, driving this reaction to the right, and producing an increase in hydrogen ions. And, to the extent we're seeing significant shifts in the pH of the oceans. And so if you consider the volume of the oceans, it will give you some idea of the volume of carbon dioxide that has started to dissolve into those oceans, that you would actually need to cause a shift in the pH, a measurable shift in the pH. So that's just a little kind of a pet thing.
So Ben's asked, so these interactions are detected only with chemoreceptors, and they are reactive to both O2 and CO2. What about lethal vapors also? Does this alter neural control?
So I'm not, unless I'm off something, I'm not entirely sure that it is that they are reactive to oxygen levels. He may be able to answer that for me, but I was very much under the impression that oxygen levels themselves played a very minor role. It was actually more the acid sensing, the chemoreceptors sensing the changes in acid environment that was driving respiration. What was the lethal vapors?
Yeah.
So what do we mean by lethal vapors? Like carbon monoxide, or maybe you can just answer that. Just give me a little bit more of a heads up about what it is that you mean by lethal vapors. So, obviously carbon monoxide is lethal. That's because it binds to hemoglobin with about 10,000 times higher affinity than oxygen does, right? And so if you think about your oxygen dissociation curve, you're not going to be able to get oxygen onto that hemoglobin molecule because it is so tightly bound to carbon monoxide, right? So it doesn't matter how low the concentration, the partial pressure of oxygen gets inside your bloodstream, that hemoglobin molecule is basically completely out of action because it's got carbon monoxide tightly bound onto it. And so there's no space for the oxygen on that hemoglobin molecule. Do we have an answer about lethal vapors yet? No? So that's carbon monoxide. I guess there's a whole collection of different vapors and gases that are going to be lethal. But some of those are actually going to be lethal because they contain lipid-soluble products that are able to pass through membranes and pass through the blood-brain barrier and directly interact with the respiratory centers themselves. Right, so those neurons inside the medulla that are driving the diaphragm contraction, they can be inhibited, right? And so, it doesn't have to be a lethal vapor. We have a massive opioid epidemic happening in the States, and there's a lot of people dying accidentally as a result of opioid overdoses. And so what's happening here is that, those respiratory neurons are controlled, and they are under inhibitory control as well as excitatory control. And so you can actually activate that inhibitory control and that inhibitory control of the respiratory function using opioids, right? So I inject exogenous opioids that acts to inhibit those respiratory neurons in the respiratory centers, and as a result, their ability to fire action potentials reduces. They're not able to send signal to the diaphragm to drive diaphragmatic contraction in order to get to get respiratory function to occur in order to lower the pressure to allow oxygen to move into the tissues. So there are a range of different ways that you can interfere with respiratory function. You're right, at the level of the brainstem. But what I found really interesting when I was in undergrad is that you have those levels at the brainstem, the neural control, but you also have really, really kind of intricate local level control around the delivery of oxygen to the tissues. But as Jamie knows, and I'm sure you probably know as well, those factors such as temperature, pH, oxygen levels, and carbon dioxide levels could also affect the dilation of the local arterioles and increasing flow of blood into the capillary bed. So this is really neat kind of local control of these cardiovascular and respiratory functions that allows us to deliver the right amount of oxygen to the tissues when they need it most. So, lethal vapors. I'm kind of curious. What's a lethal vapor?
Ben hasn't responded.
Oh, we're almost done.
Yeah.
So this is what happens when you get me talking.
Yeah.
What else?
All right. What's the difference between dorsal respiratory group and ventral respiratory group? Gamer Ethan asked me about that.
Right. So that relates to the neurons inside the brainstem.
Yeah.
Off the top of my head, I don't know. It's basically the answer. If I had my friend here who did his PhD on it, I would be able to tell you.
So dorsal respiratory group's responsible for control of initiating inspiration, whereas the ventral respiratory group is responsible for exhalation.
Okay. So dorsal responsible for inspiration, you said?
Yep.
And ventral responsible for exhalation. Okay. So, and given that exhalation is primarily a relaxation event, presumably the ventral neurons are actually quite responsible for inhibiting the dorsal group. Would be so there'd be an inhibitory network in there. Yeah. Sorry about that. It's not something I spend a lot of time thinking about, unfortunately. Well, what else do we find? What I find particularly interesting about talking about the physiology of it is thinking about the histology that Jamie would have spoken to you about last week, right? And looking at those connection points and trying to see the connection points between the tissue level structure and the cellular level structure and how that links directly to the physiology, right? And changes that we're seeing, And so if you're thinking about things like this respiratory inspiratory reserve volume, and you're talking about the forced vital capacity, we're actually looking at these physiological measures that are a direct measure of tissue level and structural level changes that are actually happening inside the lungs, right? So that's why those measures become really interesting, is they tell you about the structure of that tissue, right? They can actually give you some information about how that tissue is functioning, and changes in the function are related to changes in the structure of the cells and structure of the tissues and laying down of scar tissue and collagen fibers, etc., or whatever, decrease in elastic fibers. So that's what I find really interesting, and hopefully that's something that you're starting to get really good at, is to sort of see the connection points between the anatomy and the histology and the physiology that results from that. Nice. Well, at 4:54, maybe we say goodbye.
Yeah.
If there's no questions. So hopefully that's been enjoyable. Come on in, Jamie. And we'll say goodbye. And next week, what are we talking about?
Next week we'll be doing the digestive system. So unfortunately James can't be here next week, but I'll be here, and we can talk about all things alimentary tract. and things related to the physiology related to the digestive system. So again, if you're one of our UTAS students and you have a question you'd like us to cover, or like me to cover next week, please post it in the discussion forum on our Milo site, and I'll make sure that we cover it. Otherwise, I'll just be chattering on about the different regions and do drawings and things as we make our way through the digestive system. Haven Ammo says, thanks, it was great. Thanks very much for coming along. Really appreciate everyone coming along to these live streams. Of course, the more you come and ask us questions, the more we can engage in this process and try to help you out along the way. Damon Ethan says, thanks guys. Annabelle Claire says, thank you. Thank you very much, everyone. We really appreciate you coming along. And we'll see you next week, 4 to 5 on Thursday, Australian Eastern Standard Time. So we'll see you then. Bye.
Bye, everyone.